Hi class. I'm glad you're here. Um, yeah. Okay, question I didn't think I would need to ask, but now I'm going to ask because I'm feeling slightly paranoid. Do you get an email every week, after every lecture from me, usually about 1 o'clock in the afternoon, being like, hey, here's the link. Okay. Because I had one person today ask me something that I'm sure I've, like, sent emails over and over and over and over. And he was like, I have never in my life received an email from you, ever. I'm like, oh, I'm concerned. But okay, the people who are here know what's going on. Yeah. Okay, speaking of knowing what's going on, you do know, and again, this is not the group I'm most worried about. There are 2 papers happening. One paper for a lecture that's about something like vitamin C is good for you, so is eating vitamin C by the handful better, right? I mean, you do the research, find out. Some is good, does more equal better, right? Okay, there's that. And then there's the lab-based one, right? Where you're like, I wonder if oranges always have the same number of segments. I should check, right? I actually don't know if they always have the same segments. I should check. We're okay with what's going on here? Sweet. Yeah.
You or, I can't remember the other lab.
Yeah.
Professor's name, but can you guys, like, upload, like, what the papers are supposed to be, like, based on, like, a rubric or something onto Outnet so we can, like...
Yeah, you should have gotten an email about that. You should have gotten 2 emails about that. But yeah. Rubrics. I can get that done. It's probably soon. It might be by Monday. I have an index card full of things that need to happen today, and it's now on the list, but it's way down on the list. But yeah, if it's not there by Monday, talk to me. But yeah, basically the short version, and yes, I'll send it to you. The short version for a lecture is, hey, have at least 3 peer-reviewed sources. Like, have at least 3 peer-reviewed sources. 3 peer-reviewed sources. Show that you understand what people have done. Like, this is what's been done. Indicate some version of you can either understand numbers or read a chart or a graph, like some visual representation of numbers, whether that's in numbers, whether it's in picture, whatever. Yeah, that's that one. And then the other one is, I think that one's 2, maybe it's 3 peer-reviewed sources as well. So, science works. Science works. This is what I did. This is what I found. What does it mean? But yeah, I will send you that rubric. I'll send everybody that rubric. Oh, yeah. And you're so close, and so do a good job. But at this point, in this semester, and like, I care, and I'm going to look at every single one of your papers, and it'll be great. Just do it. At this point, just, you'll be fine. There's lots of credit this semester just for turning stuff in. 'Cause it's hard. But you can do it. And all of you here are freshmen, right? Yeah. So, I have an aunt who, the first kids she had were twins. And she was like, it was kind of hard being pregnant with them, and like having them as infants was kind of tricky. But then her next kid, she just had one at a time. She was like, this is the easiest thing ever. It's amazing. I get so much sleep. It's fantastic. All that's to say, this first semester, next year, when you can like see each other's faces and feel the joy of community or whatever else you do, it'll be great. Anyway, so hang in there, you're doing great. Papers are coming. I'll get you a rubric. You have a quiz today. Do it. The first question I got excited about, and it's this like word problem about like sloths and baby sloths. It's not as complicated as it looks. It's basically saying, do you know something about sex-linked inheritance, is what it's really asking. Anyways, on to today, are we good? Okay, so, speaking, am I missing anything? I'm probably am. Sorry. Okay. Most chromosomes in the human body and other organisms work the same way, but we're going to focus on humans for a second. They're expecting, the cell is expecting, a normal body cell, liver, kidney, spleen, eyeball, whatever, is expecting 2 copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3, the whole way down, right? The odd part is when you get to the sex chromosomes, right? So if you're male, the human body is expecting 1 X chromosome and the Y chromosome, right? And the Y chromosome is fairly small, it only has a few genes. So basically, it's expecting 1 X chromosome to do stuff. Cool. Females, you have 2 X chromosomes. And your body, though, like the human body, males are using 1 X chromosome and having it work just fine for them. Females, you have 2, but cells only are really expecting 1 per cell. So what happens is, in mammals, you've got, during embryonic development, you have 2 X chromosomes in every single cell, and 1 just turns off. It condenses down to what's called a bar body, and it's there, but it's not doing anything. So then every single cell has 1 X chromosome that's like doing stuff. Okay with that? Which can lead to some interesting things. So normally there's, for whatever protein or pigment or whatever it is you're making, like so for the color blindness thing, right, it's on the X chromosome. So long as you have some of the cells in your eyes that the bar body condenses down as the recessive one and the one that hasn't, the one that can do color sight is still there, you're fine, right? You can see, you can end up seeing the patterns like this. So cats have the same XY sex determination system as humans do. And where's my, oh, here's my step. Okay, and on the X chromosome, there is an allele for coat color, for the color of the fur. Cool, right? So color fur, and it comes in orange, or it comes in black. Now, if for male cats like this, how many X chromosomes do they get? 1, right? So every cell is going to have 1 X chromosome, and they got the X chromosome from their mom. So if their mom gave them the X chromosome for orange, that male cat's going to be orange. If the mom gave them the X chromosome that has black, they're going to be black. So far, so good? But because that gene for coat color, or sorry, for fur color, is on the X chromosome, females are going to have 2 copies of this, right? And normally in a cell, you'd be using both copies for something, and then you'd either have like a dominant situation, right, where like black covers orange, or you'd have like incomplete dominant situation where you end up with like gray or something, whatever. In this, it's on the X chromosome. And for females, 1 X chromosome in every cell collapses down. So you can end up with something like that, where the X chromosome that has the orange allele has condensed down, or you can end up with that one, where the X chromosome that has the black allele has condensed down. So then you end up with a cat that looks like that, where some cells are making orange fur, and some fur cells are going for black. Do you see this? So if you see a calico cat like this, without even looking at anything besides fur color, could you know if this is male or female? It's gotta be female, right? Because males, they only get the 1 X chromosome, so they either have to be black or orange. Do you see that? Could this female have black kittens?
Yeah.
Yeah. Orange kittens?
Yeah.
Yeah, right? It's just, you've got this X chromosome condensed down. Everyone's cool with this? Alright, so I realize I keep talking about similar things, but we're moving forward, people, I promise. Linked genes get inherited together because they're on the same chromosome. Specifically because they're close to each other on the same chromosome. So, the sex-linked genes are being inherited with sex because, sorry, not with sex, the verb, with sex, the gender, anyways. The sex-linked chromosomes do different things with males and females because they're associated on the X or the Y chromosomes, right? But there are lots of chromosomes. And each chromosome is going to have thousands of genes on it, right? Thousands and thousands and thousands, doing lots of different things. And if you've got genes that are next door neighbors on the chromosome to each other, they tend to be inherited together because they're like physically right there. They're called linked genes, and we're going to look at fruit flies and then talk some more. Okay, so this is our buddy Morgan again, the fruit fly guy, right? This experiment is very similar to ones you've seen before, but there's a twist. So he's got these gray flies, and gray is the wild type, and it happens to be dominant, and they have normal-sized wings, all right? That also happens to be dominant in this situation, okay? So he's got these gray flies, normal-sized wings, and they're homozygous for both of these dominant traits, okay? He crosses those with some flies that are black, which is the recessive trait, the mutant trait in this situation, and they have little dinky tiny little wings. They're called vestigial wings, okay? And all the baby flies that crossing have are heterozygous for both, right? So they've got the allele for gray body type, and they've got the allele for black body type, but gray shows up, right? And they've got the allele for normal size wings, and that shows up. And they have the allele for dinky wings, and that one doesn't show up. Is everyone okay with that so far? This honestly should look fairly similar to a bunch of pea plants, right? Where you're like, okay, you got two different characteristics, they're inherited, so far so good, okay? This offspring, this F1 offspring, is homozygous for both things, okay? Morgan then goes and he crosses this fly that's homozygous, heterozygous, I can say words. That's heterozygous with this fly that's homozygous recessive. Okay? We're good? You end up here, where, back up one, this dad right here, the one that you're crossing it with, he can only give sperm that have the recessive allele for body color and the recessive allele for wings. Do we believe that? We're okay with that? Now, the possibilities for what mom could give for gametes are those four up there. Right? Again, we've seen this with pea plants. Right? Now, if the two genes, like the actual genes that say how to do this, are on different chromosomes. So we've got different chromosomes, and there's a gene for body color on one chromosome, and a completely different chromosome has the gene for wings. Then we expect that all four of those combinations would be equally likely. Are we so far so good? Mom could give any one of those, it's equally likely, because they're just how they assort during meiosis. Yeah? So then you'd expect, if you're looking at, if you had that, you'd expect you have equal numbers of each type of these flies. Yeah? Okay. If, however, the body type, the body color, and the wings are next door neighbor buddies, they should get inherited together, right? So you have, like, let's see here. if there's a chromosome in this fly that's one chromosome and the wing type and the body type are right next to each other, that means there's a chromosome here that's this one, and the other chromosome has the two recessives. Do you see that? So if you're expecting, if the genes are on the same chromosome, you're expecting that you see those first two types of flies, right? But then you wouldn't see these mixed up type of flies, right? This fly would look like mom, this fly would look like dad. These two flies, the mom has a gray body and big wings, and this is gray body, small wings, and that you wouldn't expect. And the dad has black body, small wings, and this one's black body, large wings. Do you see what's going on here? Do you want to see the results? Drum roll, it's going to be anticlimactic. All right. Feel mildly bugged. I'm hilarious. Sorry, I didn't do it on purpose. Okay, sorry, thanks. Thank you for the pity chuckle. I actually do appreciate the pity chuckles. I'm going for like comic bronze here, people. Okay. Is this person right, or this guess right here, where genes have different chromosomes, so you're expecting equal numbers in each of the 4 types? That ratio does not look like 1 to 1 to 1 to 1, right? Okay, so I don't like the idea of these being on different chromosomes, because if they're in different chromosomes, they should assort independently, right? But looks like these things are staying together usually, but not always. So it does kind of look like they're on the same chromosome, because like the ratio is about 1 to 1, but not quite 0. Right? Any guesses what might be happening? Or should we just chug along? Okay. We're gonna keep chugging along a little bit. So Lynette does this experiment and lots like it. Morgan's finding out, they're like, okay, cool. Body color, wing size are normally being inherited together in these specific combinations. And they're called parental phenotypes. All right? And it has nothing to do with like dominance always inherited with dominant, recessives always inherited with recessive. That's not true. You could actually have where you have a parental fly that has like a black body and large wings. If that's happening, you're expecting those two to be inherited together. So Morgan's looking at this and he's saying, okay, these genes aren't assorting independently. So they've got to be on the same chromosome. Right? If they're on the same chromosome, then they're coming together in the same package. So you'd see a picture like this, which so far makes some level of sense, right? You've got a chromosome here. These are our F1s, right? After the first cross, they're the ones that are the second cross, where you have the mother is heterozygous, right? She's heterozygous, but with both wild types on one allele because they came from her mother, and the male is homozygous recessive. And so you end up with, the dad gives the same one in both cases, but you either end up with this type here, and this offspring looks like mom. Not genotypically, but phenotypically looks like mom, right? And this offspring over here, which is this one plus this one, looks like dad, right? Is everybody fine with this? So those are called parental phenotypes. So, yeah. Taken together with some Mendel stuff, we're going to figure some things out. If the offspring has the phenotype that matches one of the parental phenotypes, it's called a parental type. Okay, because it looks like mom or dad. We're talking phenotypes here, people. If offspring have non-parental phenotypes, there's a new combination of traits that neither parent had. Right, so each parent can have some, but neither of them had that combination. That's called a recombinant type, right? And basically, well, we'll get to it later. If genes are on different chromosomes, you're expecting a 50% recombination rate. We're going to see this here. Back to Mendel and his peas, right? You've got this parent up here that's a yellow round parent. It's heterozygous, all right? You've got this parent over here that's homozygous recessive, right? And when you cross those two, you end up with this ratio here. Are we okay with why you end up with this ratio? Okay, we've crossed that a long time ago. We're good. You end up with this yellow round P. Does this yellow round P look like either mom or dad? Don't worry about genotype, we're talking phenotype, physically. Yeah, right? This parent is yellow and round. Cool, it looks just like mom. Oh, I can see it in your little P roundness, right? Okay? Does this P look like either the mother or the father? Yeah, this one's also green and wrinkly, right? This one's yellow and wrinkly. Is mom yellow and wrinkly? Is dad yellow and wrinkly? No, right? So it's a recombination. So it's a recombination of the yellow aspect from mom and the wrinkly aspect from dad. See that? And then this green round P does the same deal, where mom gave it round and dad gave it wrinkly. Sorry, dad gave it green, and it's a new combination. Okay? If genes are on different chromosomes, you're expecting them to recombine about 50% of the time, right? Half the time they come together, half the time they don't, just because of how the homologous chromosomes line up, how meiosis happens. If they're on the same chromosome, you expect them to be linked together. Fair enough? But they're not linked perfectly. Back to our fruit flies, even if they're on the same chromosome, sometimes they have those recombinant types. So yeah, offspring that don't look like the parents are called recombinants or recombinant types. Anyways, so Morgan's looking at this and he's like, okay, somehow the chromosomes have to break up and like rearrange themselves. Have you seen chromosomes breaking up and kind of rearranging themselves? Oh, you have. You guys are amazing. We'll show the video. Oh, okay, gosh. I'm going to show the video and then we'll come back to pictures. Online people, this is in your notes, but we're going to watch it right here.
Phase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of two non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from two parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes.
Right? Okay, so we've seen this before. Great. That then explains this chart. It's in your book. I stole it from your book. I realize it looks intense when you look at it at first. You're like, oh, too many fruit flies. It's not terrible. Yeah. So this is actually where we just were a minute ago, right? Where you end up with this heterozygous fly, right? And then when you cross this heterozygous fly with the homozygous fly, like this, the homozygous recessive fly can only give the sperm, cool. But the options for what the mom's giving are either this allele that has these genes linked, this one again linked, or if crossing over happens, you also have these crossing over ones. Yeah? Concerns about this? People are okay? Then when you look at the offspring, you can look and you can be like, okay, so we have a whole bunch of this parental type, a whole bunch of this parental type, a few of this recombinant type, and a few of this recombinant type. And if you do the math on this, because you're this dedicated kind of person who's counting fruit flies, you can be like, all right, we have like 2,300 fruit flies total. And of those, there are 391 of those that are recombinant types. So crossing over between, somewhere between where the gene for body type was and the gene for wing type, some crossing over had happened between those 17% of the time. Do we see that? Because you have to have it break and flip in between those two genes to make that happen. So 17% of the time that happens, right? Thought experiment. If you were trying to put genes on a chromosome, and you want them to always be inherited together, to be like the bestest little gene buddies, where do you put them? Do you put them like, so you're gonna put one gene, another gene, and you're gonna put them like, you want them to stay together forever, besties for life. Do you put like one way out here and one way out here, or do you jam them as close together physically as possible? As close together as possible. How come? So they don't get separated. Are we okay with this? This is a terrible example, but I'm going to go with it because I thought of it this morning while talking to the other class, and they were unimpressed, but who cares? We're going to keep going with it. If I have this chromosome, and actually has two sister chromatids, but this is just the one that's going to do crossing over, and I have this chromosome, the homologous chromosome, it has another one too, but I only have two hands, and they come next to each other, right? If I'm trying to get this with something over here, I can have a crossover event occur here, 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 anywhere along here, and they'll flip. If I have two, if I'm trying to get these, okay, gosh, here we go. If I'm trying to get these shapes to switch, I can have a crossing over event anywhere between here and here. If I want these little black lines to switch, the only place for that event to happen is right here, right? It's really easy for things on this end and this end to cross over because it belongs anywhere here. But for this one to cross over, I have to peg it exactly there, right? The insight that apparently you all just thought of like in 2 minutes was a big deal and was kind of impressive. And apparently undergrads just like had the ability to make this insight because Alfred Sturvant, the name I forgot on Wednesday, was one of Morgan's students, and he worked that out. And this is the quote. I love this quote. I suddenly realized that the variations in the strength of linkage, right, so how come sometimes things are like inherited together, but other times they're only sort of linked, already attributed by Morgan to difference in the spatial separation of the gene, offered the possibility of determining sequence in the linear dimensions of the chromosome. So he's saying, I figured out that because they come in different strengths, I can figure out what order they're happening on the chromosome. I went home and spent most of the night, to the neglect of my undergraduate homework, in producing the first chromosome map. I love this. He went home, he's like, yeah, and he drew a map of the fruit fly chromosome, which is beautiful. If we had more time this semester, but we don't, we would spend a fair bit of time making maps of chromosomes, because why not? But we're going to look at it just briefly. You don't have to do anything with this, just look at it. And like, I mean, I guess learn something. If you have so many parts here, we're just going to look at this, lots of things happening, but we're just going to look at this one. If you're looking at the recombination frequency of different genes, and you see that, okay, the recombination frequency between A and B, so normally A and B are inherited together, but 13% of the time A and B are not inherited together, okay? And you see, all right, genes B and C, they're inherited together most of the time, but 6% of the time they're not. So the recombination frequency is 6%. All right? And we see that, okay, genes A and C, they're inherited together most of the time, but 18% of the time they aren't. Do you see that? Alfred Sturvant's insight into this was, hey, if I know those recombination frequencies, I can get the order that they show up on the chromosome. Because, just don't look at this yet, but because of this, which ones of these are the closest together, meaning they're the least likely to split apart? Which one's the least likely to have a recombinant type? We're okay with these guys, right? Only 6% of the time they're not inherited together. Usually they're inherited together, but 6% of the time they aren't. Whereas these ones are usually inherited together, but like basically 20% of the time they're not. So you think, okay, these ones are close, these ones are farther away, and these ones are somewhere between these two. So you realize, okay, A and C have to be the farthest apart of any of these two things, and B has to be somewhere in the middle, and B is way closer to C than it is to A. You can actually do this with like all the things. If you're super into it, you can like make the map of everything. It's cool, but we're gonna just move on. But realize, if you want to do this, sign up for genetics. Yeah. Also, so if you've got a gene that's like way up on that end of the chromosome, and another one that's way down on this end of the chromosome, yeah, they're physically on the same chromosome, but they don't really behave like they're linked, because crossing over almost always happens at least once. So they're physically linked, but genetically they're unlinked, because they don't always have to be inherited together. Does that make sense? Great. All right. Yeah, that's. I was just saying that. I should read my notes. Okay. Last bit of things to know. I will let you out a little bit early for your quiz. These heritable factors, right? This genetic stuff. It's living on the chromosomes. That's great. It's definitely there. If chromosomes start doing funky things, that is going to lead to odd things happening. And this is not a value judgment on like, if you have weird things happening in chromosomes, cool. Biologically, it's odd, all right? Plants are fine with having all sorts of weird chromosomal things happen. Animals have a hard time, especially mammals, have a hard time with chromosomes getting funky. Something that can happen is called non-disjunction, where pairs of homologous chromosomes don't separate the way they're supposed to. So you end up with a gamete that doesn't have enough, or you end up with a gamete that has too many. So it can happen in a couple different ways. It can happen in meiosis 1 or meiosis 2. So if we're up here in meiosis 1, and these homologous chromosomes are supposed to be pulled apart, but in one case they're not pulled apart, like this should be happening, but no. You can then end up with a combination, something like this. We're gonna follow this one on the left first. These get pulled apart. Here they're gonna go through meiosis 2, and then instead of what you're supposed to have is you're supposed to going through meiosis, you're supposed to end up with 4 gametes, each with 2 chromosomes. If non-disjunction went down here meiosis 1, you end up with 4 gametes. Each of them has the wrong number of chromosomes. Half of them have 1 too many, right? Because you split here, you end up with 3 here, it should be 2. And half of them are missing 1. Do you see that? If you have non-disjunction happen in meiosis 2, the homologous chromosomes split up fine. but the sister chromatids didn't separate. So then you end up, this one over here is doing just fine, right? Split apart, split apart. These guys are great. But these ones over here, you have one gamete that has one too many chromosome, and one gamete that's missing a chromosome, right? So then when there's some gamete that has like one too many chromosomes, and it meets up with a gamete that has the normal number of chromosomes, and you get the organism, that organism has one too many chromosome in every single one of its cells, right? Or the reverse, where you're missing a chromosome, the gamete that that has the normal amount comes there, every single cell is going to be missing a chromosome. Fair enough? Theoretically, I don't know that it's ever been seen, but theoretically, if there's a problem with both the egg, so the egg has like one too many of a specific type of chromosome, and the sperm has one too few of a specific type of chromosome, you could solve it, but like that doesn't happen. But theoretically, but it doesn't happen. Because non-disjunction isn't super common, but neither is it super rare. So far so good? You can see this video. Watch over here on the screen. You're watching most chromosomes get together and line up and separate, but watch for one chromosome that is not participating appropriately. It's gonna be right over here. Okay, they're getting lined up, Watch for it. Everybody else has decided to pair. Do you see it? It stayed over there? That's a problem. Do we need to watch it again? Okay. This is the most exciting video you'll see in this class today, so, you know, the bar is low, but it is exciting. Okay, so they're trying to condense and get organized, line up at some sort of metaphase plate. They're working on it. This chromosome has not joined this blood part. This one stayed here. It did. Yeah. You see that? Now, there's going to be funky numbers of chromosomes in the thing. Once fertilization has happened, you can end up with either, if there's been non-disjunction, you can end up with what's called a monosomic zygote, meaning there's one particular chromosome that only has one type of, or you can have a trisomic zygote, so one that has 3 copies when you should only have 2. Yeah! Yes, because I skipped stuff. Okay, so, it's called aneuploidy, which is just where the gametes had come together and the chromosome numbers were off. So there's an abnormal number in the offspring. And yes, I skip stuff, sometimes I shouldn't, but here we are. So far so good? I'm going to skip a couple things here because I skipped them for this morning's class because I put so many slides in that we're going to have to see a little bit of this on Monday. But one thing to see, polyploidy is when an organism has more than 2 complete sets of chromosomes. So triploidy is where it has 3 complete sets of chromosomes, or tetraploidy, where you have 4 complete sets of chromosomes. Humans do not do this, like, ever. Plants, though, plants do polyploidy all the time. They're cool with it. Animals don't do this. Okay, we're going to skip these slides here. We're actually going to get back to some chromosomal alteration stuff. And we'll get done in like 3 minutes so you have some time for your quiz. Genes are on chromosomes, right? They're telling the body how to make all these proteins. This is great. Chromosomes have thousands of these. So if you're saying missing an entire chromosome, that's bad news for the organism, right? Like it's not easy to survive. Yeah. So, people heard of Down syndrome? Heard it called trisomy 21? it's technically trisomy 21. It's where an individual has 3 copies of chromosome 21. I want to show you this picture. So this is a karyotype. So that picture right there is looking at the chromosomes of this little girl. All right? So look at it, and we see chromosome 1, there are 2 pairs. So far, so, sorry, 1 pair, so 2 chromosomes. So far, so good, right? Chromosome 2, chromosome 3 has 2 pairs, right? So on and so forth. Remember, they're, with the exception of the sex chromosomes, they're numbered in size. Okay, so we get moving down. We see chromosome 20, there's a pair. Chromosome 21, there are 3 of them. You see that? Chromosome 22 have 2 of them, and then she has 2 X's. Right? Huh. What do you notice about the size of, say, chromosome 21 relative to most of the other ones? It's very, very small. Is it 2 options? 2 options. Tell me which one you think, and then we'll talk about which one's actually. Trisomy 21 is the most common trisomy that humans know about, that we seem to have. So there are 2 options. One option is there's something about chromosome 21 that just makes it so that it's the one least likely to participate appropriately in splitting apart, right? That it's just the one that non-disjunction happens to. And that all the rest of the chromosomes are better at this somehow. The other option is chromosome 21 and all the other chromosomes are all equally likely to have non-disjunction happen. It's just you never see trisomy 1 because what you see is miscarriage, because the organism cannot survive not having chromosome 1, because chromosome 1 has so many important things. Any guesses which one it is? Yeah. All right. So all these chromosomes are equally likely to forget to show up, like we were splitting today, right? But you don't see trisomies 1 or 2 or 3 just because the developing organism doesn't get far enough. A lot of miscarriages are like this. A lot of what are called chemical pregnancies are like this, where in chemical pregnancy, the woman doesn't actually know she's pregnant. So, fertilization happened, but it stopped. There's not the information needed to keep going. So, honestly, it just looks like a really heavy period, where it's like, oh, we started, but we missed a step there. So far, so good? There are some other trisomies that happen that are survivable. They're all on little chromosomes with the exception of sex ones, which we'll talk about later. Concerns, questions, comments? We're good? Take-home messages, do your quiz, I'll send you emails. By Monday, you should hopefully get a rubric. Heritable factors show up on chromosomes. X-linked stuff do funky patterns. Genes that are next door neighbors get inherited together. Great. Yeah.